Today we're going to talk about species and site-specific genome editing in complex bacterial community. And what we are facing is that a lot of microbes are still uncultured and limiting the application of traditional genetic method to them. So the author tries to find a new way to do these things. And what they have done is the new approach is a generalizable strategy for editing the genomes of specific organisms in the wild-type microbes community. And they use two tools to help them. One is ET sequence and the other is a DART system. And the result is that they can do species and site-specific edit in soil and invertebrates microbial communities. The problem of classical genetic method faced in is that is genetic mutation and observation of phenotype outcomes. And to do the classical genetic editing, we have to isolate species, lead to three disadvantages. First is many of microbes have not been isolated in laboratory. And the second one is genes involved in interaction between microbes mostly unexplored. And third is isolated microbes will hide their wild-type biology. It will be more useful if we can do precise genome editing in wild-type communities. And so they have a new approach to do it. And the first one is ET sequence, and it's a method shows that which species are easy to do gene deliver and editing in a microbe community. And we want to know which gene deliver method works best for which microbes. And the third is the result of ET sequence will help us to do next step, the DART system, which can do target gene transposition. And here's the method to do the ET sequence. And how to do it? First, we transform marina transposon into a micro community with three methods by conjunction, electroporation, and natural transformation. 3 ways. And second step is we use ET sequence machine to determine insertion location and quantity in each host, and by enrich and sequence the conjunction between the inserts and host DNA. And third step is we use low-depth metagenomic sequence to quantify the abundance of each species in the sample. And finally, we calculate insertion sequence data relative abundance. Then we can get the insertion efficiency of each method in different species. In this example, the blue strand is most suitable to electrophobic, as we can see here. And the result of the ET sequence based on this strategy, we compare the insertion efficiency in 9-member synthetic soil community, and we can identify 5 species that are easy to do gene deliver, as we can see here. And each method are measured 3 times. And although didn't detect insertion in other 4 species, which may be because their abundance are just too low, below ET sequence limit of detection. And here are the results of the 5 species and their best gene deliver method. And as you can see here, the first one, the highest one is conjunction. So the best deliver method is conjunction. And the second one is conjunction also. And the 4th and the 5th one, the best way to deliver is electrophoration, because the one is the highest. And the second step is develop a CRISPR transposon system. And why the team used CRISPR technique to do this research is because the RNA guide CRISPR-Cas7 transposase could provide the ability to both delete the function of target gene and deliver custom genetic cargo in organism. And the previous plasmid limitation is they are low efficient delivered with complex micro communities or even beyond E. coli due to their multiple plasmid. So the solution is that we use the ET sequence result to identify the best deliver way to deliver the system. And then we also design an all-in-one plasmid to avoid multiple plasmid. And the all-in-one plasmid also is almost the same as normal CRISPR system, but with a barcode for ET sequence and integrate all the genes that need it. And as you can see here, there are two kind of dot system. One is SH dot system and the other is VC dot. And we have to compare these two transposition efficiency and specific. And by using ET sequence, we can find out that SH dot approximately 10-fold more column position insertions than VC dot. And over 92% of the selectable column obtained using SH dot were off target. And the VC dot, no detectable off target insertion among the 1,586 independent insertions. And so although the SH dot system is very high efficiency, but the specificity is very low. So here the author choose VC dot as their CRISPR system. As you can see here, the VC dot system has only one insertion, and the SH dot, although it also have this insertion, but you can see here there's a lot of off-target sequence down here, so the specificity is very low. And application of CRISPR transposon system for bacterial community. And by combining the two results of ET sequence and the dot system, the author chose this as their research because of the abundant member shown by ET sequence. As you can see here is 71% of the whole microbes. And the conjunction was used to introduce the VC dot vector into the community because of is the highest. And with multiple guide RNA specific to two locations in the K and this strain genomes.
The research team do counter selection. To check this method is really useful. They delete PYRF and cultured under growth inhibitor to show fitness measured by ET sequence. They first do tagging and tracking of mutant fitness in microbiota. The mutations contain a barcode VC dart transposon, disrupting a gene of interest, PYRF, and an internal control locus, referred to here as a safe site due to its predicted fitness. Safe site guide means that it will insert the position doesn't affect bacteria growth. And this picture showing the use of ET sequence to quantify the fitness effect of a VC DART mutation of interest, measured as a ratio of mutant of interest rate normalized to safe-side mutant rates before T0 and after T final, outgrowth of the mutant-containing community. And if the line goes like this, it's a positive fitness. Research team predict that disruption of targeting PYRF in K-strand would facilitate fast growth in the presence of growth inhibitory 5-FOA. It means that if the bacteria grow without PYRF, can survive in 5-FOA. The edited community was grown in the presence of 5-FOA, and ET-seq was used to quantify the effect of this condition on the fitness of PYRF mutant in the community. And here's the result. Results showed positive fitness. Fitness of PYRF mutant under 5-FOA treatment as measured by the ratio of PYRF to safe side read. And here is the result. It goes like positive as expected. ET sequence detect higher fitness of PYRF mutants relative to safe side mutants in the presence of 5-FOA. The combined efficiency of VC DART editing and ET sequence detection is thus able to analyze gene fitness and function directly in microbial community without disturbance caused by traditional mutant selection requirement. And the research team do application in synthetic soil community to test this measured. They're using both positive selection and negative selection to K-strand and P-strand to isolate each species from the community. They use VC DART inserting antibiotic resistant gene AADA or BLA to disrupt PYRF gene. And picture B is the result. Different gRNA can select different bacteria in community, and the results show that the mixed culture after selection lead to 99% purity of targeted species. For these three, they only have K-strand, and these three only have P-strand. So it shows that the purity is really high. Selecting species via antibiotic marker might influence the bacterial community. So we can test selection target organism via given metabolic ability, instead of using the antibiotic to select. Integrating lactose assimilation pathway into P-strand within a community that can utilize lactose. However, the growth of Rostonia species was also observed. And we will explain why happened this later. A selecting strand in the minimal media contain lactose as solo carbon source. So if the strand don't have leg Y and leg Z, they can't utilize lactose. So if they can utilize lactose, it means that they have these two genes being inserted to the bacteria. The research team do shotgun metagenomic sequencing and show that only one pair rates of integrated cargo in one replicate. So they're suggesting that the off-target integration was not the major source of Rostonia species persistence. Next, the research team do application in an infant gout consortium. The infant gout sample was mapping via a reference genome, and only E. coli subspecies showed editable within a community in the infant gout sample. The detected E. coli strains are members of B2 and D phylogroups. Research team using VC DAR targeting the clinically relevant site and integrating antibiotic resistant gene ADDA and BLA. After selection, E. coli subspecies 2 and 3 were enriched. The dually identified via metagenomic sequence. Research team to ET sequence showed that specific integrating in complex natural community was achieved. ET sequence was used to map insertion location. Results show that 199.2% of insertion occur within 20 base pair of the expected insertion site within E. coli subspecies 2 and 3. And here is the conclusion and discussion. The research team demonstrated that the use of ET sequence, a method allowing mapping and quantifying targeted genes, and DART are an all-in-one vector contain CRISPR-Cas transposon system. Combine ET sequence and DART to isolate targeted strain from a complex community using an antibiotic marker or create a metabolic ability. And the future improvements in both editing efficiency and ET-seq limit of detection will allow further sensitivity and enable assaying the fitness of multiple genes in one experiment, as well as understanding the impact of such genes on the fitness of other organisms in the community. And here is all of our presentation. Thanks for your listening.